बिस्मिल्लाहिर्रहमानिर्रहीम। स्टूडेंट्स, हमने स्टार्ट की हुई है मोनोक्लोनल एंटीबॉडीज। तो हमने उसके थ्री स्टेप्स पढ़े थे फॉर द प्रिपरेशन ऑफ मोनोक्लोनल एंटीबॉडीज। द फर्स्ट स्टेप वाज द प्रोडक्शन ऑफ और द सिलेक्शन ऑफ द बी lymphocytes. The production of B lymphocytes can be initiated by adding a antigenic mixture into the animal model, and the spleen of that animal model will result the B lymphocytes. Secondly, in the second step, we require the stem cells or bone marrow cells, or the known as myeloma cells. The myeloma cells will be attained from the stem cells, from the bone marrow cells of the human, or it can be synthetically synthesized. The synthesized stem cells will become hybrid with the B lymphocytes in the presence of PEG media, polyethylene glycol media. This union can be processed in a medium known as hat medium. Hat medium will require the supply of HGPRT enzyme, and that HGPRT can only be supplied by the B lymphocytes. It cannot be supplied by the myeloma cell. So myeloma cell cannot grow indefinitely because they cannot partner this enzyme. And the B lymphocytes, as they are not immortal, so they will also die after a specific period of time. Only the cell which will become a hybrid by making a union of myeloma cell and the B lymphocytes, they will be able to partner this enzyme due to the presence of B lymphocytes, and they will remain immortal, or they will grow indefinitely due to the presence of the myeloma cells. So they can be checked or verified by using ELISA technique. In the fourth step, we will have to culture those monoclonal antibodies or those hybridoma cells which we have developed for monoclonal antibodies. So we have to culture them so that they can grow in the indefinite model. For example, they can grow in colonies known as single cell colonies are known as colon, and the single B lymphocytes colon is known as monocolon. Only one B lymphocyte that can result into a colon will be monoclonal. So we have to grow in the medium. So there are actually two methods to grow or the culture the hybridoma cells. One of the method is the selection of a suitable media in which we can culture the hybridoma cells, and the other method is the selection of animal model in which we can culture the hybridoma cell. First of all, we will discuss about the culture media which can be used for hybridoma cell culturation and its ultimate growth. So the culture medium actually used for the culture of hybridoma cells are normally supplied with some hormones, with some insulin, transferrin, ethanol, amines. These kinds of nutritional factors are required in which hybridoma cells can be cultured to form monoclones. So the commonly used mediums which contains these supplemented materials are Delbaco modified eagle medium, Scovos modified Delbaco medium, Hams F12, and Roswell Park Memorial Institute 1640 medium. These four are the common medium which are used to culture hybridoma cell and to get the colonies known as clones of the monoclonal antibodies that can be injected into the other animal model to verify that whether they are producing the specific response or they are not producing the specific response. Second method will be the culture of the hybridoma cells into a animal model. So it will be called as inoculation of hybridoma cells into suitable host, and then purification of monoclonal antibodies from the spleen of that host. So if we can add the hybridoma cells in an animal model will produce a specific response or antibodies because they will divide in the spleen of the animal, and animal can produce monoclonal antibodies. So hybridoma cells can be injected into lab animals or animal models so that they start producing monoclonal antibodies. So the attained hybrid monoclonal antibodies, which are of the abundant in number, attained from the spleen of the animal, can be frozen and can be stored for the future use. So monoclonal antibodies from the host animal is extracted, and then they can be purified from the spleen cells, from the cells and the precursors of the blood, by using some techniques, chromatographic techniques. For example, two chromatographic techniques. One is ion exchange chromatography and other is antigen affinity chromatography. They can also be isolated from immunoassay techniques. For example, a radial immunoassay technique, in which we use a radial, a source of the radiation that will result in the immunization, that will result in immunes which are actually producing some variable scintillating waves. And then there is a method of precipitation also, in which we can use immune precipitation. So these methods can be used to isolate the monoclonal antibodies which are being grown in the spleen cells of the lab animals, so that we can purify alone monoclonal antibodies from those cells. So here is a diagrammatic summary. We have prepared a monoclonal antibodies, and then we have injected it into a animal model. For example, we have injected into mice. Mice is known as a model of murine model, M-U-R-I-N-E, murine model. So if we are injecting antibodies into mice, it will be known as a murine model injection. So from the spleen of this murine model, we can attain the B lymphocytes. On the other hand, we have culture of myeloma cell. For example, we have a cultured synthetic stem cells. Those stem cells will be actually used for the production of myeloma cell. For example, we will use myeloma from those stem cells. In the polyethylene glycol medium, we can make a hybridoma of the spleen cells, B lymphocytes, and of myeloma cells. And further addition of a selective medium, for example, that was hat medium, we can only grow the hybrid cells. For example, these two colored union is hybridoma, and a single color are of the individual cells. So hybridoma can only selectively can be grown in a specific medium, for example, a hat medium. So we can attain these hybrids in a glass, and these can be propagated. For example, they can be propagated through PCR technique, and the propagation, or they can be propagated by a specific technique, which is microbiological assay like technique. Also they can be propagated in that, or we can use a culture media. Here are the two examples. We can use a culture media to propagate it, or we can use an animal model to propagate these antibodies. For example, in a culture media, we will select a culture media. We have discussed four: Delvaco, Icecovo, Delvaco modified media, a Hams media, and similarly a last of an Institute 1640 medium. In these medium, we can grow it, and those can form colonies of the monoclonal antibodies, and that antibodies can be extracted from that culture medium. Secondly, we can inject it into the animal model. For example, gain a murine model. So murine model can be injected with hybridoma cells, and from the spleen of that murine model, we can attain monoclonal antibodies that will be known as murine antibodies. The major problem which we have actually now discussed is about the selection of mouse model to produce the monoclonal antibodies. If we are using a mice genome to generate B lymphocytes and myeloma cells, if we are taking myeloma cells or B lymphocyte cells from the mice, and normally we have taken spleen cells from the mice, we have developed stem cells of the mice, and then we have united to make them hybridoma cells. As those are generated purely from the mouse genome, so they are called murine antibodies. And murine MMABs means murine monoclonal antibodies. Murine MMABs have some problems because they are specifically different from the human genome. Mouse genomes is that is producing antibodies from the same antigen have different coding as compared to the human which are producing the antibodies from the same antigen. So the end product murine, known as murine monoclonal antibodies, having a different amino acid sequence cannot be administered to humans because they can be allergic to the human, because sensitivity response can initiate if we are adding genetic makeup which is different to the human. Human will produce antibodies against that monoclonal antibodies. So it will result in the killing of those monoclonal antibodies. Secondly, murine monoclonal antibodies, because they are of the genome of the mouse, so they are actually short-lived. As the life of the mice is short-lived, similarly these murine ABs will be short-lived. So they can be removed, they will be removed from the circulation fairly quickly, and we cannot result a proper propagation inside the human body. These two critical problems urge us that we can use only those B lymphocytes which are generated by humans, and use only those stem cells which are of the source of human body, or they are synthesized from the genome of the human body. This type of solution have a problem. For example, hybridomas which are made by B lymphocytes originated from humans, and stem cells which are synthetic stem cells of the human genome, they are unstable. These hybridoma cells are unstable. They are not propagating for longer period of a time, and they get isolated in the normal conditions. Secondly, when we are using human to get the B lymphocyte, so we have to add some antigens inside the human body. The human body will not produce B cells. For example, it will produce a response, but that response will be in the blood, not in the spleen of the human. The spleen will contain only those antibodies which are being propagated, or they are of a large number, or for having a second or a third exposure. So we cannot extract those kind of B lymphocytes from the blood, as we have the techniques which can extract the B lymphocytes from the spleen of the animal. So human and monoclonal antibodies are less possible. Secondly, B cells, if they are produced from the human source, they will not multiply when they are added to a human, as human will not produce human B cell against the human tissues. Human antibodies will not result in the immunogenic response inside a human. Human B cells cannot be secreted. And lastly, there are ethical issues also, that we cannot immunize a human with antigen which can result some diseases. So it is ethically not a good go that we will go with humans. So there are safety issues also. So there are multiple monoclonal antibodies which are from the murine antibodies, and as the murine antibodies generate immunogenic response also, so we need to add some antibodies that can suppress that immunogenic response along with the treatment of the disease. So it is a very difficult way to treat with murine antibodies. There is another solution that we can use a hybrid monoclonal antibodies. Hybrid means we can use a chimeric monoclonal antibodies, or chimeric monoclonal antibodies. Chimeric monoclonal antibodies have the variable. We have discussed the beta B lymphocytes. They have the three regions. One is the binding region, and the two are the one normal. The lowermost is the constant region, and the uppermost is the binding region. So if we can make a monoclonal antibodies by taking a constant region from the human from the human basis and human genes and the variable region from which epitope is actually existing and B lymphocytes are being generated, it can be selected from the mouse genome. So we can reduce the immunogenic response by multiple factors. For example, for 75%, if we can use a constant human region. So the hybridoma cell line generating murine antibodies against desired epitope, and they can be used as starting point. Then we can use a reverse transcriptase PCR enzyme, in which we can make DNA copies which are actually complementary DNA copies. With the reverse transcriptase PCR, we can use messenger RNA that will result into the coding of cDNA, a complementary DNA. So we can make constant region by producing a genetic messenger RNA which is producing the constant region of the antibodies of the human by reverse transcriptasing it and then making its multiple millions of copies by using PCR technique. So recombinant DNA technology will allow the cDNAs for the variable region to be combined with DNA coding of human constant region. So cDNA can be developed for the murine and DNA of the human can be used for the constant region. So recombinant DNA can be developed in which the variable region of the mice, murine, and a constant region of human can be designed. And then when we combine this recombinant DNA with the myeloma cells, using the expression vectors, for example, this can be of the mice or this can be of the human myeloma cells. So as they are synthetic, they are non-sensitive or they are non-immunogenic or less immunogenic. They are normally tolerated well if they are of the synthetic source. So a synthetic source myeloma cell derived from the human genome can be hybridized with the recombinant DNA of monoclonal antibody, which have a constant region of the human and the variable region of the mouse. So it will result in the variable region with an epitope that is from the murine, and secondly the constant region which is of the human. So the major part of the monoclonal antibody will be from the human source, and it will be less immunogenic and will interact least with the human receptor to elicit any therapeutic response and interact well with the disease citing species to enhance their therapeutic effects. The example which is of the chimeric MMABs or chimeric and MMABs which are used for non-Hodgkin lymphomas. Way past 2000, in 2005 or 6, it was actually registered as the drug, the MAB is rituximab. MAB आ गया आपने बोलना है. So rituximab is an example of chimeric MABs which are used for non-Hodgkin lymphomas. But we have seen that when this rituximab was administered to the patients to treat this kind of a cancer, there was some immune responses also generated against these MMABs. So the only variable region also initiated some immune response. So we have modified it into some better way, which is known as humanized monoclonal antibodies, humanized MABs. Humanized MABs are similar to chimeric. For example, they have a variable region of the mouse and the constant region of human genome. But we have actually also replaced those low binding and high binding sites in the variable region which are other than the epitope by the human sequences. If we can exchange the murine sequences with the human sequences, other than the epitope, we can develop a antibody with majority of the bases originated genes or bases originated from a human source. So it will further reduce the immunogenicity. So complementary determining region or CDR, this complementary determining region is actually the epitope. So complementary determining region can be inserted inside a whole human based MAB. So it will result in the chimeric MAB to be called as humanized MABs. But there is a major problem with this humanized MAB. Humanized MAB is containing only a CDR, a complementary determining region from murine ABs. They have the ability to reject those CDRs, so it can lose its specificity. It cannot bind to the antigen then again. So a specificity can be lost, a binding activity can be lost. A lost binding activity will result in a failure to the humanized MABs. So there is ideally, instead of using only CDR, we should use some a bit low density, binding of the murine region, murine residues that to be introduced inside the variable region so that they remain functional and intact. A humanized MAB with CDRs and some bases which are attached to the CDR of the murine residues will result in the more stable preparation. So that stable preparation example used for the breast cancer treatment is of the humanized MABs are trastuzumab. To minimize these side effects, we have a constant struggle that we can prepare some fully human MABs. So there was a solution to this. Instead of using humans to sensitize with an antigen and produce B lymphocytes against the humans that cannot generate the response or cannot generate the B lymphocyte in a human, we can use some mice which have the human strains, which has human type of cDNAs inserted so that they can make B lymphocytes of the coding that are used in the human coding. So these mouse are actually developed, or animal models are developed, known as xenomouse strains. Xenomouse strains are the mice but having the B lymphocytes, or their immune system work according to the human genetic makeup. So they are known as xenomouse strain. So when these mice are immunized with the antigen, the immune response will constitute of the bases which are used for human B lymphocytes. So a fully human antibodies will be isolated from the spleen of the xenomouse. So when we made hybridoma of those and B lymphocytes with the humanly generated myeloma cell, synthesized human myeloma cell, they will result in fully human MABs. So ideally, this is actually the example which is used entirely for entirely human MABs and are used as a immunosuppressant drug. Vastly used immunosuppressant drug is ideally MABs. Ideally MABs. So they can use as a immunosuppressant drug, and they are of entirely human amino acid basis, so known as fully human MABs. This figure differentiate between the murine, chimeric, humanized, and fully human monoclonal antibodies. In the murine, which is indicated by the genetic sequence of the mice in the red coding, so all the constant region and also the variable region. This variable region and this constant region are of murine or the mice. So it is murine MABs. And if we replace this variable region with the variable region to stand with the murine, but the constant region and the heavy region of the variable region can be replaced with human. So it will be known as chimeric. We have reduced the, so there we are actually seeing in a diagram 4 types of MABs. A murine MAB constitute of a amino acid sequences attained only from the mice. A chimeric MABs that contains amino acid sequence of the variable region from the murine and from of the normally constant region from the human amino acids. And in a humanized, we have actually replaced those variable region of with the humanized amino acids. So humanized amino acids can replace majority of the variable region. Only the CDR region and some bases which are of low density, they can be used as a base attachment to the CDRs. And then we have developed a mice which is known as xenomouse. Xenomouse can produce all the antibodies with fully humanized amino acid sequences. So those antibodies, B lymphocytes generated from the xenomouse will be actually a human monoclonal antibodies, but they will be generated from mouse and they can produce immunogenic response in the body for those monoclonal antibodies. And monoclonal antibodies can generate indefinitely. From here, we will move to the therapeutic application of monoclonal antibodies. So here are multiple slides that are with the trade names. Trade names means they are actually registered and they are being used for the therapeutic response. These are the monoclonal antibodies which are used for multiple diseases. For example, for the cancer diseases, lymphocytic leukemia, cutaneous T-cell lymphoma. For example, to use as immunosuppressant to treat rheumatoid arthritis. Similarly, to treat infections. Similarly, they can treat some tumor cells of the bones. They can treat metastasis of the bone, osteoporosis. They can treat osteoporosis. The main interest is in the last slide. We have recently used some monoclonal antibodies. For example, tocilizumab. Tocilizumab was prescribed rapidly during the COVID-19. It was prescribed rapidly during COVID-19 along with some steroidal preparations, for example, dexamethasone. So dexamethasone and tocilizumab was concurrently administered. They were administered in the first phase of the infection, for example, within the first 9 days of the infection. So they can, this monoclonal antibody have the property that it can reduce the rapid respiratory decomposition. It will reduce the decomposition response which our respiratory was facing due to the COVID-19 and also significantly increase the markers of the inflammation. Secondly, remdesivir was administered during viral stage of the COVID-19 and it reduced mortality rates up to 70% when it was administered during the early viral stage. For example, in the incubation stage. In the incubation stage, the remdesivir was used to decrease 70% of the mortality rate that was actually without the remdesivir, it was with COVID-19. And then new inventions were rapidly coming into the science with some antibodies. For example, there was a COVID-19 which is known as regenerated or second generation or third generation COVID-19. So COVID-19 which is regenerated COVID-2 was called COVID-2. It was treated with casirivimab and imdevimab. These monoclonal antibodies were available vastly normally from the Roche. Roche produced these two monoclonal antibodies. And then bamlanivimab and stacivimab. These are of the third generation COVID-19 monoclonal antibodies. there is a list of the monoclonal antibodies which have been used in the treatment of COVID-19.